Hello, everyone, and welcome to today's keynote webinar titled Targeting T-Cell Malignancies with CAR-Based Immunotherapy, Challenges and Potential Solutions. This webinar is a part of the third annual event in the Immuno-Oncology virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. All right, now let's get started. I'd like to now present today's speaker, Sunil Raikar, Assistant Professor of Pediatrics, Emory University, Aflac Cancer and Blood Disorder Center, Children's Healthcare of Atlanta, Junior Director of Graduate Studies, Molecular and Systems Pharmacology, Graduate Division of Biological and Biomedical Sciences, Emory University. For a complete biography on our speaker, please find it on the left of your screen. Sunil, thank you for joining us. You may now begin your presentation.
Thank you, Kaylee, for that kind introduction, and good morning, good afternoon, all our to our audience today. So, my name, as Kaylee mentioned, is Dr. Raikar. I'm at Emory University, and I'm a physician scientist with a background in pediatric oncology, and so on the clinical side, I take care of children with pediatric blood cancers, and so my research kind of is also focused in that area. And so today I'm going to talk to you about one of our main research interests in the laboratory, where we target T-cell malignancies with a CAR-based approach. So I have no conflicts of interest to disclose. First, I just want to give you an overview of the talk. I'll briefly give you a review of what T-cell malignancies entail, then talk briefly about what CAR T-cell therapy, or chimeric antigen receptor T-cell therapy, involves. Discuss the specific challenges in using CAR-based therapy in T-cell disease. And then I will talk about 3 different approaches that we've used in our laboratory to target this disease using a CAR-based approach. So that includes studies involving the natural killer cell line NK92, CRISPR-Cas9 editing-based approach, and then finally talk about some of our gamma delta T-cell based work. Finally, I'll go over, just briefly review a couple of the clinical studies that have now been published in the last couple of years of using a CAR-based approach in T-cell malignancies, and finally, I'll give you a summary. Okay, so to get started, T-cell malignancies. So these, as we all know, T-cells is one of our important immune cells, and so sometimes there's mutations that develop that leads to a T-cell malignancy. And so this encompasses a heterogeneous group of diseases, each reflecting a clonal evolution of dysfunctional T-cells at various stages of development. And so if you look at it, there's a different subset for both pediatric T-cell malignancies as well as adult T-cell malignancies. On the pediatric side, the main 2 end up being T-cell acute lymphoblastic leukemia, or TALL, and then its lymphoma version of this, which is called T-cell lymphoblastic lymphoma. And a minor subset of T-cell malignancies in the pediatric side end up being peripheral T-cell lymphomas, or also known as PTCLs. And within that, there are several different subtypes, the most common, at least in pediatrics, being anaplastic large cell lymphoma. Whereas on the adult side, yes, there is TALL and T lymphoblastic lymphoma, but peripheral T-cell lymphoma becomes a much larger component with very many more subtypes aside from ALCL. But as I mentioned before, my clinical focus is in pediatric oncology, so I take care of children with TALL. So most our lab's goal is really focused towards curing TALL. So briefly to get into TALL, this accounts for about 15% of childhood malignancies, so it's probably the 3rd most common after BALL and AML, and about 25% of adult ALL cases, with B-ALL obviously being the more common type of ALL seen both in children as well as adults. As I mentioned before, it is the most common form of T-cell malignancy seen in children. The survival in pediatrics, we do pretty well with intensive chemotherapy, with now survival ranging in the 85 to 90% range. This has been primarily done through intensifying chemotherapy regimens. And so most of our patients we treat with chemotherapy alone, and only once they relapse do they then need a bone marrow transplant. It's slightly different on the adult side, with survival being much worse, with survival only being 50% at 5 years. But as I mentioned, once these patients relapse, it is very difficult to treat them because they often are very chemo refractory. So if you look at the survival for relapsed TALL, it is much more dismal. So in the children, it's only about 20 to 25%, whereas in adults, it's less than 10%. Now, a way, as I mentioned, to cure these patients is to offer allogeneic bone marrow transplant. But unfortunately, to get to transplant, you need to get into clinical remission. And often, as I mentioned before, it is very hard to get these patients remission because they are very chemorefractory. And so if you're able to get to transplant, you do have a 50% survival chance in children, about 30% in adults. But most of these patients, unfortunately, cannot proceed to BMT because they're not able to get a remission. So clearly, there needs to be newer approaches that are developed to target this disease. Another thing that we've kind of looked at, in terms of approaches, when you look at relapsed TL and you do when we've done these genomic studies, it's really a very heterogeneous disease. So there is not a single target to go after. And so even though we've attempted some small, targeting certain pathways with small molecule inhibitors, it really hasn't panned out to be better than any of our previous chemotherapy approaches. And part of this, as I mentioned, is because genomically, it's a very heterogeneous disease with numerous different subgroups. So all this leads to kind of what we're going to talk about today. And so I mentioned chemotherapy is very difficult to use in the relapse setting. And so one of the approaches we're trying to use is to use immunotherapy. Until this point, it's really, has not been successfully used in this disease. So we're trying to develop better approaches. If you talk about cancer immunotherapy, in general, you can break it down into probably 4 different categories. Initial work involved using cytokines, so you use recombinant cytokines to activate your immune system. Monoclonal antibodies has been part of our therapy regimens, rituximab, for example, targeting CD20. More recently, over the past decade, immune checkpoint inhibitors have gained a lot of prominence, more so in the solid tumor side with using nivolumab or pembrolizumab. But what I'm going to focus is on an approach called adoptive cell therapies. So known as ACT. So what is adoptive cell therapy, right? This is basically the transfer of specific immune cells into patients to then target and kill their cancer cells. This can be done in 2 forms. One, an autologous approach, where cells are expanded from the patient's own blood, or what we call an allogeneic approach, where cells are expanded from a healthy donor's blood. And so there's 4 different steps involved. First, you obtain the blood either from the patient itself or a healthy donor. You then isolate PBMCs, so peripheral blood mononuclear cells, and then expand the specific immune cell of interest. In most of the cases, this is T cells, but there are studies that use NK cells, gamma delta T cells, or NKT cells. So there are other different types of immune cells that have been explored now to use for adoptive cell therapy. Between the second and third step, either at this point is where you may use genetic modification of the immune cells to enhance specific targeting of cancer cells. And so this is where, when we talk about CAR T cell therapy, this is where the cells are used to undergo lentiviral transduction to incorporate a specific protein called ACAR, which I'll talk to on the next slide, to then modify these T cells. Then they are able to then specifically identify and kill cancer cells. Once this modification is done, these immune cells are then infused back into the patient, where then they go ahead and target and kill cancer cells. So I did briefly mention the term CAR. So CAR stands for chimeric antigen receptor. And what this is it is an engineered T cell receptor which can bind specifically to a target surface antigen on CAR T cells. And the most common form of this involves linking a single chain variable fragment, which is derived from a monoclonal antibody, as you can see in the figure, figure below, um, where you take the variable heavy chain and a variable light chain from the monoclonal antibody, link them with a peptide linker, and then incorporate that into the CAR sequence. So that ends up being your antigen recognition sequence. You then have different components of the T cell receptor, primarily the CD3 zeta intercellular signaling domain that's then connected to the scFv. And so that was what we called a first generation CAR. As we developed this technology further, we realized that you need to incorporate more co-stimulatory domains into the structure to optimize T cell activation. As we all know, T cells require two signals to undergo activation. And so by incorporating these co-stimulatory domains, which could either be from CD28, 41BB, or OX40, we then were able to design a more optimal CAR structure. And so that when you incorporate one co-stimulatory domain, it's a second generation CAR. When you incorporate two, it's called a third generation CAR. And for the most part, second generation CARs have shown the most success on the clinical side. And so that is what we have proceeded with in our design of the CAR. So just to kind of mention some of the clinical success that is in. So most of our clinical success has been seen in targeting B cell antigens. And primarily the first four FDA-approved CAR T cell therapies were targeting a protein called CD19, which is seen on normal B cells, but is also seen on B cell leukemias, right? And so now we have four FDA-approved, as I mentioned. They're called Kymriah, Yescarta, Tecartus, and Breyanzi, and they cover a spectrum of B cell malignancies ranging from BALL, diffuse large B-cell lymphoma, mantle cell lymphoma, as well as other large B-cell lymphomas. Now, one of the toxicities of CAR T-cell therapy is that it does not necessarily distinguish between tumor and normal cells. It's really just identifying a surface marker on a cell, right? So in the case of CD19-directed CAR T-cell therapy, it also kills your normal B cells because it identifies that as a target cell. And so this leads to something called B-cell aplasia, where you just wipe out your normal B cells and you lose your humoral immunity and are not able to produce antibodies. But luckily, we've been able to treat this with a drug called intravenous immunoglobulin, which is a pooled collection of antibodies. So most of these patients that undergo CD19-directed T-cell therapy receive monthly infusions of IVIG, and this allows them to not have significant B-cell mediated immunodeficiencies. Some of the other side effects seen with CAR T-cell therapy include cytokine release syndrome, which is CRS, which is basically a hyperinflammatory T-cell state. And then you can have this form of neurotoxicity. So I'm not going to get too much into that because I want to transition really into talking about targeting T-cell disease with CAR T-cell therapy. So as I mentioned, right, in CD19-directed CAR T-cell therapy, you'd lose your normal B cells. Now, we do have a solution to that because we can able to treat these patients with IVIG. We're not so lucky on the T-cell malignancy side. So when you target a T-cell marker with CAR T-cells, you'd also end up having T-cell aplasia, which can lead to significant immunosuppression. And so that could be life-threatening. And so that's a problem, right? And so really the challenge in targeting T-cell malignancies is the lack of a tumor-specific surface antigen on cancerous T cells. So ideally, we would just want to be able to develop a CAR T-cell that can target the cancerous T ALL cells, leading to anti-tumor activity. But unfortunately, if you have a T cell marker that's also present on normal T cells, it's first hard to develop a CAR T cell product because as you're expanding this product, it might cause targeting of each other, a process called fratricide. So then that makes it challenging to develop an effective CAR T cell product. As I mentioned before, if you target normal T cells, you can have this process called T cell aplasia, which leads to life-threatening immunodeficiency. And then finally, another challenge that can happen with this product is, when we typically collect immune cells from a patient, we're able to separate out the T cells from the rest of the immune cells using T cell markers such as CD3, CD28. Unfortunately, some of the cancer TALL cells also express these markers. So when you're trying to isolate healthy T cells from a patient with TALL, you may end up with a product that also has contamination with some malignant leukemia cells that then subsequently may undergo, when you transduce this population, may start expressing a CAR, and this by then ends up blocking that epitope, causing relapse. And so that can also be a challenge where you might end up with a contaminated CAR T cell product. So those are the main three challenges that are involved in using this technology for T-cell disease. But our group and several other groups have used several different approaches to overcome these challenges. And so I'm going to just go through them one by one. So for this concept of fratricide, where basically the CAR T cells target each other, there's multiple different ways that groups have gone over it, and including some approaches that we've used ourselves. One is by using genome editing. So we'll talk a little bit about CRISPR-Cas9 editing, where you basically edit out the target antigen for your T cells and then modify them to express a CAR. That way they no longer cause fratricide. You can target antigens that are downregulated. There's a certain number of antigens as soon as the CAR is expressed, just have downregulation, and so they no longer leads to fratricide. You can target antigens which have limited expression on T cells, so can only target a portion of T cells. Then you can include more genetic engineering by a TET-off expression system, where you don't downregulation expression of the CAR until you expanded the population. Some groups have used something called protein expression blockers, where you are able to block expression of the surface protein and thereby prevent fratricide. And then alternatively, instead of using T cells, you can use other types of immune cells, such as NK cells or NK92 cells, which have a different surface antigen profile, thereby giving you the ability to pick surface antigens that are no longer expressed on your immune effector cell. For T cell aplasia, again, some of these approaches, as you look, will overlap with each other, but you can target antigens with limited expression on T cells. Therefore, you're not wiping out all the T cells. You can use some transient methods such as mRNA electroporation or AAV vector delivery, which leads to transient expression of the CAR. So it's not a permanently expressing CAR T cells. So you can then have a limited effect where it's not a long-standing effect. You can use cells that have a shorter lifespan, such as NK or NK92 cells or gamma delta T cells. So then it's not a persistent effect, therefore not leading to long-standing T cell aplasia. And then other approaches is using safety switches or suicide genes, where you just switch off the CAR after a certain amount of time. And then finally, you can bridge these patients to an allogeneic hematopoietic stem cell transplant, where you're just completely replacing the immune system and wiping out the CAR, so then you don't have a sustained T cell aplasia. And then finally, to talk about product contamination, really, when you're talking about product contamination, it really negates using an autologous approach. So here, you basically have to collect cells using an allogeneic approach, where you're collecting cells from a healthy donor that is not the patient. So if you typically use alpha beta T cells, your regular T cells, there's a high risk of causing graft versus host disease. So you might have to do some genome editing, such as editing out the T cell receptor using TRAC locus or TRBC locus editing. Alternatively, you can use NK cells, or you can use gamma delta T cells, which have a different T cell receptor and are not known to cause GVHD. So these are multiple different approaches that have been used by multiple groups. And so we had kind of put out a review paper a few years back where we looked at all these approaches. And so when you try to combine them into a Venn diagram, you can see that some of these approaches overlap and kind of are able to address multiple issues at once, right? And so basically what we did is we listed all of these different approaches that have been explored by different groups and put this in this one figure. So for the next portion of this talk, I'm going to specifically focus on 3 different approaches that we have tested in our lab here at Emory, and kind of give you an idea of how much progress we've made. So the first one, as you can see, one that overlaps all 3 of them, is using basically not a T cell based approach, right? Using a different immune effector cell. In our case, we use natural killer cells, and specifically this natural killer cell line called NK92. Okay. There we go. So why natural killer cells, right? This is a cytotoxic lymphocyte of the innate immune system, and it accounts for about 7% of your total lymphocyte pool, but really it has a limited lifespan. So it doesn't have this ongoing persistent persistent effect, and so can avoid some of these issues such as T cell aplasia. Now, in terms of fratricide issues, it has a very different surface antigen profile than a typical T lymphocyte, and so a CAR NK cell approach is more feasible to target T cell malignancies, as long as you're able to choose a specific antigen that's not expressed on the surface of NK cells. And initially we used this NK92 cell line, which has been out there It's an IL-2 dependent cell line that was derived from a patient initially who had NK-like lymphoma, and it's been used as a good preclinical tool to test for NK-based approaches. Now, in terms of our target antigen, we chose to go with CD5. This is a pan-T cell marker that's expressed in most T cell malignancies. In terms of its role, it's a negative regulator of the T cell receptor, has been shown to have a role in protecting against autoimmunity. And back in the late 80s and early 90s, it has been used to study as a tumor-targeted antigen, specifically against T-cell malignancies. And there's a couple of papers using an immunotoxin conjugated CD5 monoclonal antibody-based approach. So and there has been some responses shown in cutaneous T-cell lymphoma. So it seemed like a good target. But more importantly is, as I mentioned, it's not expressed on natural killer NK92 cells. So it allows us to overcome the issue of fratricide since it's not expressed in NK cells. So, when this was when some of my work from my fellowship work, when I joined my mentor, Dr. Spencer's lab, they had an interest in using a different CAR design. So instead of using the traditional SCFB, which is derived from a monoclonal antibody, we were using something called a VLR, which stands for a variable lymphocyte sequence and receptor. And so it is meant to move to the next slide. So it stands for variable lymphocyte receptor. So what this is, it's a single chain structure that represents the functional unit of the adaptive immune system in jawless vertebrates, so lampreys and hagfish. So it's very different from your typical immunoglobulins that you see in humans. And the reason we were looking at this approach is, one, it is a much smaller single chain structure. So in terms of genetic engineering, it's much easier to incorporate into a CAR structure, whereas the way we design SCFVs is we artificially take out the variable heavy and variable light sequence and then join them by a peptide linker. But sometimes the orientation may not be appropriate because it's in a different structure now. Now it's as a single chain structure. So that was one of the reasons. The other, beyond the CD5 approach, is we thought that VLRs could potentially recognize novel targets, including proteins and carbohydrates that were not recognized by mammalian antibodies. So hopefully, we were hoping that we could potentially identify novel targets. And then because of the evolutionary distance between mammals and lampreys, it basically expands the tumor antigen repertoire. So there's a potential to develop a whole new system of VLR CARs. So that was the idea. But first, as proof of concept, we wanted to go with a target that we already knew. Just to kind of briefly go what the lamprey VLR structure is, it's a single chain structure, but its sequence identifying structure involves several leucine-rich repeats, which are assembled in very unique formation in each. And so basically, that's their VDJ system, where the different combinations of these leucine-rich repeats allows for variability in antigen binding region. And so they have a whole repertoire that goes from 10 raised to 15. So it's a very extensive potential identification. But the reason we went with this with the CD5 is there was a publication that was already out there back in 2012 that had already isolated a VLR that specifically identified CD5. So it seemed like it would be a good tool to use as proof of concept to see if this VLR CAR approach worked. And since it's targeted CD5, we felt like this could be an interesting project to take on to develop a CAR for a T-cell malignancy. So we created what we called a second generation CAR. As I mentioned, that means there's one costimulatory domain. So ours included a CD28 molecule, but instead of a typical SCFB from a CD5 antibody, we use this CD5 VLR sequence as our antigen recognition sequence. We then generated lentiviral vector using a 4-plasmid system, and then went ahead and modified NK92 cells. Before that, we did that just for proof of concept. We did something called a JERCAT activation assay. So if you know, JERCAT is a T-cell leukemia line that's kind of used as a preclinical tool to look at T-cell function. And so we transduced a JERCAT T-cell line with this vector that encoded the CD5 VLR CAR. And as you can see, as we increased our MOI, which is multiplicity of infection, there was higher activation. We believe that this was self-activation caused by the CD5 CAR interacting with CD5 on neighboring molecules. Now, since Jurkat cell is a T cell line, it does not have any cytotoxic function, so it really doesn't cause fratricide and kill each other, but it could cause activation. And so that told us that our CAR was really functioning because there was a clear link between the amount of virus used to the degree of activation. And this activation decreased over time because since the cells were constantly activated, this led to activation-induced cell death. And we were able to demonstrate the CAR using a Western blot. So, sorry, before I was trying to, I almost skipped that part, but the more important part was going back to using this NK cell approach, right? And so we transduced, for this, our initial experiments showed that the NK cells are very difficult to transduce, which is not uncommon. Natural killer cells or other innate type immune cells are indeed very resistant to lentiviral transduction. And so what we did is we created this dual vector that expresses GFP, green fluorescent protein, as well as the CAR. And then this allowed us to select for the positively transduced cells. So we had about 2 to 5% transductions with the vector, and we saw GFP, but able to expand that flow sort and expand the cell line to create a NK92 cell line that was specifically expressing our CD5 VLR CAR. So this kind of just shows some of that work where we had very low transduction, but once we were able to sort and expand it, we had very high transduction. This Western shows good expression of the CD5 VLR CAR with a decent copy number. Okay, so then to test it, we use a flow cytometry-based cytotoxicity assay where we labored our target TLL cells with a membrane dye called PKH26. And so then this allowed us to separate both our effector cells from our target cells, and then we stained the combination. We cultured these cells in different effector to target ratios for about 4 hours, and then we stained the cells with a dead cell marker 7AD before flow cytometry analysis. And so this allowed us to quantify any killing that the CAR has done. And so this was the first time we had demonstrated that a VLR-based CAR can kill a leukemia cell line. And so as you can see in the figure on the left here, as we increase the amount of CAR NK cells, you had much higher killing, especially when compared to the naive, unmodified NK92 cell line, which did not have that much specific killing against the TLL cell line. So this was really good proof of concept that a VLR CAR could be used. But again, still, this was just a concept of a VLR CAR. The next step we felt like was we do need to compare this to a more traditional scFv-based CAR, because that's really what that's derived from more a mammalian antibody. And we wanted to see if there's any better effect on that. So we basically designed a CD5 CAR, a scFv-based CAR, using based on a sequence monoclonal antibody clone called 865, which was published back in 1994. And we took the variable heavy and variable light chain and linked them by using a G43 linker. And again, we did the same process where we created this bicistronic vector that also expressed GFP that allowed us to flow sort and expand an NK92 cell line. And so when we compared this VLR CAR to scFv CAR, even though they both had similar activation, what we saw was that the activation initially seemed to be better with the VLR CAR. And so we felt like this was better, but they had a very similar trend where the activation kind of just died over time. And this is, again, showing our process of sorting and then expanding that line. And so here we can see both of these NK92 cells express the VLR CAR or the scFv CAR. If you can notice here, the VLR CAR, it's smaller in size because that single chain of a VLR is much smaller than the traditional scFv, which we always thought would be an advantage in terms of engineering down the road. But unfortunately, when we started doing side-by-side in vitro cytotoxicity experiments, we saw that the scFv-based CAR had more of an advantage over our VLR CAR, with a difference about 10 to 15% higher killing at all ratios. And this seemed to then, the difference panned out when we did an in vivo study against a Jurkat leukemia-based mouse model where we injected NK92 cells 4 times. As you can see, the VLR CAR seemed to have some survival advantage over the naive one, but that really didn't pan out long term. But clearly, the mice that received the more traditional CD5 scFv-based CAR had a significant advantage in survival. So This basically, just to kind of summarize this part of the work of our talk, we were able to construct a CD5 CAR using both the VLR and scFv. Both of them showed good in vitro activation. However, when we compared the both of them head to head, it showed that the CD5 scFv CAR had superior killing both in an in vitro model, but more so in an in vivo model. So this portion, then the next step is to transition into primary NK cells. So for this next part of the talk, I'm going to talk about our next approach, and this is using CRISPR-Cas9 editing. And so what we decided to, at least we're planning to do, was to first target the target antigen. So in this case was CD5. That way there's no fratricide. But then in order to make this overcome this issue of product contamination, you do need to create an allogeneic CAR. So that we're trying to achieve by using TRAC locus editing, which basically is TRAC stands as a gene for the T cell receptor alpha chain. So by disrupting that, you don't allow for assembly of the T cell receptor, and thereby you can prevent potential GVHD, making this a more allogeneic product. So, okay, so for this, just briefly, I'm going to mention what CRISPR-Cas9 genome editing is. Basically, it's a two-component system, basically consisting of a single guide RNA, or sgRNA, which is a 20 base pair region that can specifically identify a sequence of a gene that needs to be edited, right? This is then paired with a Cas9 protein, which is derived from a bacteria that contains two nuclease domains which cause a double-stranded break. And so by using this system, you can really specifically target a specific gene, leading to gene editing. And the way this is delivered is using a method called electroporation, where you deliver an electric pulse that disrupts the cell membrane in causing formation of pores, which then allows you to then deliver this two-component system to cause editing. And so we did some of this work. We first started with Zircat cells. This was one of the graduate students at the time, Dr. Fleischer, who worked alongside me in Dr. Spencer's lab. And we were able to generate a CD5 negative cell line, Zircat cell line. When we then transduced with our CAR, we're able to show that the edited cell line had a much higher expression of CAR compared to the non-edited CD5. So what we felt that was happening is the CD5 CAR intracellularly was binding with their endogenous CD5, leading to a decreased expression of the CAR on the surface. So we felt like this would have a dual advantage in that it would have higher expression of the CAR as well as causing decreased fratricide. Unfortunately, some of our primary T cell work is, what we realize that electroporation tends to be pretty harsh on primary T cells. And at that point, we started to look at different potentially alternative methods to electroporation to see. And so this has been reported pretty well. The poor viability that can be seen with electroporation, although techniques have really gotten better over the past few years. This is just some data that was published from the first CRISPR engineered T cell trial that was published back in 2020, where you can see after 2 days after electroporation, fewer than 20% of the transfected cells remain. So it really does cause a decrease in viability. However, in terms of alpha beta T cells, if you're able to let that culture grow for long enough, you are able to eventually expand those cells. But along this time, since we are at Emory, we have a close collaboration with Georgia Tech, and they use a lot of bioengineering methods to kind of overcome some of these challenges. And I had an opportunity to collaborate with Dr. Todd Selchuk, who is an expert in microfluidics and has a specific interest in delivering molecules into cell. And so he had this microfluidic device called a VECT device, which stands for Volume Exchange for Convective Transfer. And basically what this device does is it has a series of ridges which allows the cells to be compressed, and this mechanical compression then causes membrane pores to open up, which then allows for molecules to be delivered into the cell. And so you do this a series of times, and basically by causing these pores, you deliver material into the cells. And so they had done some work kind of just looking at this approach just to deliver, just in terms of cell delivery. And we had this opportunity to collaborate where he wanted to test to see if this could be used to deliver CRISPR-Cas9 editing molecules. And I had a specific interest in trying to do this because we wanted to, use this editing approach to deliver specifically develop CAR T cells, edited CAR T cells. And so we decided to collaborate to kind of see if this worked. And so initially they had just done this on some other cells, not specifically T cells. And so when we started this collaboration, we just kind of wanted to specifically look at this first in some Jurkat T cells and then primary T cells. And one of the bigger things, one of the biggest advantages this has over electroporation is, one, you can deliver large size cargoes up to a large number of cells at once. And so you can deliver it to about 100 million cells, about 5 million cells per minute. So you can really get to that number pretty high. But the biggest advantage was you have very high cell viability post this mechanoporation. And about 95%. Whereas for electroporation, there's definitely hit, and you always are somewhere in that 40 to 60% range once you come on the other side of it. So the thought was that this delivery system could cause really few off-target damage, and then the cells would expand better post-edit. And so this work was done by one of graduate students in Dr. Soltek's lab, Tongyu, who just actually defended last week. Should get her PhD soon. Let me see if I can show you a video of how. So this is what the device looks, and this is kind of one of those ridges where you can see the cells flowing through. I'm going to play this video, and you can just see how the cells flow through this ridge, and that's when they're under. Okay. So that's kind of a sense of how the cells just flow through the device and then get compressed, and that's when we deliver material into the cells. And so first, our efforts started. We started with Jurkat T cells. Just as I mentioned, it's a good tool to use to look at T cell biology, and we're able to show pretty decent CD5 knockdown efficiency with it. However, the amount with electroporation was always a little bit higher in terms of the delivery, but what we did see is that the post electroporation, the viability dropped compared to the mechanoporation. And so the proliferation, when you took into account for proliferation and viability, there was a much higher efficiency. So looking at the first work here, if you can look at panel B, we had about 70% versus only 30% for the editing efficiency. But if you look at panel C, when you expand these cells, electroporation really, the cells took a hit for the first 5 days. However, the mechanoporation, those cells grow pretty well post-expansion. So if you took both factors into account, we saw that we had a much higher number of CD5 edited cells a week post this. However, there are numerous factors involved in this, and I'm not going to get too much into it, but I'm just going to mention on the next few slides, where you can change the flow rate, gap size, the spacing of the ridges, the thickness, as well as the number of ridges, and that allows us to optimize the device. So we then adopted this approach for primary T cells. Again, this then involved an activation step. So we activate the cells on day -2, and then 2 days later, we did this mechanoporation to deliver the Cas9 along with the guide RNA. And then we quantified this about 5 days later. And this work was published last year. And again, there was a lot of work that ended going into optimization of this VEST-based device for CRISPR-Cas9 editing. This gets more into bioengineering, so it really was work done by the Georgia Tech group. But just to mention, kind of look at some of this, you can, for example, looking at the gap size, there was definitely a difference with the in terms of if you used, there was a sweet spot for the primary T cell. So if you'd use 5 micrometers, you had a much higher efficiency compared to 6. Also, the pressure of delivery seemed to matter, with a 6-bar delivery leading to better delivery. And so, again, there was a lot of work done on their side trying to identify different features and then optimizing that. One of the things was this focusing of how you deliver the cells, as I mentioned, the spacing, there was using a gutter, and then the number of ridges as well as the ridge width. And so once we had that all optimized, we then kind of created a workflow to incorporate this mechanoporation device or VEC-based editing into our CAR T cell manufacturing workload for developing edited CAR T cells. So the way this worked was we activated the cells on day 0, then we performed editing about 24 hours later. This was then followed by transduction with a lentiviral-based vector expressing encoding a CAR, and then we allowed the cells to grow, and then we assessed for knockout and CAR expression on day 9, followed by a killing assay. And since this was proof of concept, we first used the more traditional CD19-based CAR just to see if the cells work. And so that was our first initial testing that we did. And as you can see, we had very good editing. I mean, we had about 50% editing among of these cells using this device, which was pretty good. And then about 35 to 40% CAR expression post-transduction. In terms of killing, it seemed to work really well, where they had a much higher killing than our control unedited population. And also, when we looked at the T cell phenotype, it seemed to be more in the naive central memory phenotype, which is what's thought to work really well with CAR T cell efficacy. So this was the first instance of using this editing platform into making a CAR T cell, and so we published our first paper on this. And so now we're working towards kind of transitioning to our real interest, which is doing a dual edit and then creating a CD5 edited CAR T cell. And so we have made a couple runs at looking now as a dual edit, and we clearly can see that you are able to do. This is editing both CD5 as well as the T cell receptor, and we're able to show that using this device, using different kind of combinations of the rich space and rich width, as well as the pressure, we're able to optimize and get about a 30-40% dual edited population. Now, obviously, to kind of then transition this into an allogeneic product, we would have to get more into 100% range, but it doesn't, from where we seem to kind of hit a plateau around 60 to 65% with these edits. So there will definitely need to be a selection step in process where we can then select out the alpha beta positive TCR expressing cells before this can be then converted into an allogeneic product. And this is a similar approach that's been used by other groups as well. So just to summarize the second portion, we've shown that CD5 edited CAR T cells can show increased expression of CD5 CAR, especially in the JERCAT T cells. We've then used vector mechanoporation to CRISPR-Cas9 edit, and it seems to have a higher overall yield compared to electroporation. And then we've successfully incorporated this device into a gene-edited CAR T cell manufacturing workflow. And we're able to generate double. So the next step really for us is to then now create these double edited T cells and then transduce them with a CD5 CAR to create this potential allogeneic CD5 CAR using this approach. And so that's where this project is currently. So as I transition to the next time, I just wanted to kind of mention a couple of things, as far as, we talked about CD5 CAR editing. And so as the field, as you know, multiple groups have been working on this simultaneously, as it happens with all CAR T cell targets. And, we've really looked at editing it out because to preventing fratricide, and there's other groups that have done similarly because other groups have shown that CD5 blockade can really enhance the CAR T cell or T cell cytotoxicity. And really some group, UPenn is using this as a pan approach by knocking out CD5 to really enhance the anti-tumor activity. However, in parallel, the Baylor group had put out this paper where they showed that, that when you're able to expand CD5 CAR T cells without editing, because there's downregulation of CD5 as soon as you express a CAR. And initially, when this work was coming out, we were kind of looking at it closely while we were working on our NK cell-based approach. But they were able to publish this, and then I'm going to briefly mention some of this. They've been able to move this approach using an unedited CD5 CAR T cell into a clinical trial. So I kind of just want to leave you with that. And it did seem to be that was, the limited fratricide was dependent on the co-stimulatory domain they used, because they did see expansion with the CD28 based approach, but when they switched it over to 41BB, they saw significant fratricide and were not able to use that approach. But they did go ahead and move this into a clinical trial using a CD5 CAR, using a CD28 co-stimulatory domain. So I'll briefly mention that towards the end. So just in the interest of time, I think I'm going to move to our next portion, since we're about, we got about, yes, I don't want to miss out on these last few slides. So the next approach we use is something called gamma delta T cells. Again, using CD5 as well. And the reason we use this called gamma delta T cells, there's a focused interest in our lab. These are a subset of T cells that you see in your peripheral blood. It's about less than 5%, but the biggest difference compared to our traditional alpha beta T cells is that they recognize their targets in an MHC independent manner. So they do not require antigen presentation. And so they have a minimal risk of GVHD. So they become an ideal tool to create an off-the-shelf allogeneic therapeutic. Now, they do have a very limited lifespan, which can be a double-edged sword. So on one end, you cannot get a sustained effect, and so you need multiple infusions. However, this does prevent the risk of any off-target toxicity, which becomes important when you're targeting a T cell antigen, because you don't want to have long-standing T cell aplasia. Now, we have a very well-established protocol in our lab where we can expand these gamma delta T cells from blood, and specifically that is the V delta 2 V gamma 9 subset that's seen in blood, and we're able to expand this to numbers that are then clinically relevant for adoptive cellular therapy. Just to mention, this is our expansion protocol. We use a combination of zoledronic acid and IL-2, and then we have an alpha beta T cell depletion step that's incorporated at day 6, and we're able to get close to a 50 to 100-fold expansion from what we start with. So if we start with about 100 million cells, about 2 to 5 million of these are gamma delta T cells, then you'll be able to expand them back up to that 80 to 100 million at the end of day 12. So it's a pretty robust protocol that's been well established in our lab for over the past few years. Now, going back to the CD5 CAR work, since Baylor had published its data showing that there's CD5 downregulation, we tried to do this in an unedited approach. And so, however, it's similar to the NK cells, gamma delta T cells being more of an innate-like immune cell, also is very difficult to modify using a lentiviral vector and has very low transduction efficiency. So then that led us to use a more transient approach using AAV vector mRNA electroporation. And we're able to show good CD5 CAR expression. There's definitely downregulation. However, we were able to show good killing, but the viability of the cells using an electroporation-based method is pretty low, and so it's hard to then work with these downstream. So we're trying to incorporate now the VEC device into our editing of this device, because there does seem to be that there is significant fratricide between these cells, even though there's downregulation. So it's a combination of the electroporation approach as well as having just fratricide from the CD5 expression. So we're trying to create a CD5 expressing gamma delta T cell using a VEC-based editing approach. And so that's kind of in the work right now. But as we were having some struggles with editing, one of the important things with gamma delta T cells is they have inherent anti-tumor properties. So at baseline, they're able to recognize markers that are seen on leukemia cells through NKG2D and DNAM1 receptors. So these identify what we think are called stress receptors. And so TALL cells, or T cell leukemia lines, have these NKG2D ligands such as ULPB1 to 6, as well as they have these DES pathway receptors such as FAS and TRAIL-R1 that also can be recognized on the receptors their ligands can be recognized on target cells. And there's also some killing that's mediated by the gamma delta T cell receptor itself, which can recognize phosphoantigens, which are then cause conformational change in molecules called butyrophilins. And so some of the work, when we looked at it, we felt, yes, we would ideally like to create a CAR T cell, but since the cells themselves kill, we started working on a project where we combined them with chemotherapy. And this work was done by one of our former graduate students, Dr. Jamie Story, where we combined gamma delta T cells with a chemotherapy drug called bortezomib, and just wanted to see if this combination could lead to increased killing. And as you can see on this slide here, when you treat leukemia cells with chemotherapy, there is upregulation of these markers such as ULBP2, 5, 6, which are again recognized specifically by NK cells, but as well as by gamma delta T cells. And so when you use a combination approach between these two, we had much higher killing when we combined this gamma delta T cells by themselves along with with chemotherapy. And so we've now expanded this work to using different drugs such as azacitidine and venetoclax, which are drugs that are now being used more in the relapsed refractory TLL setting. And our early mouse data actually does look promising, where we're seeing an increased survival benefit of using chemotherapy in combination with gamma delta T cells. So we're eventually hoping that this can turn into a clinical trial where we use allogeneic unmodified gamma delta T cells in combination with chemotherapy as our initial proof of concept of using gamma delta T cells, while we continue to work on the engineering of gamma delta T cells, which is a little bit more challenging than your traditional alpha beta T cells. So just to kind of summarize this portion of the talk and I'm just going to have a few more slides after this, expression of CD5 CAR did result in downregulation, and they were cytotoxic against TLL cell lines. However, there does seem to be significant fratricide, and so there needs to be continued optimization of CD5 editing and CAR expression in these cells, and then we will proceed with in vivo validation. But in the interim, we've also notified that gamma delta T cells at baseline have higher killing against TLL cell lines, and this can be maximized by combining this approach with chemotherapy. And we have an ongoing in vivo xenograft study that's currently undergoing and has shown some promising results. So I know we just have a few minutes. So I'm just going to go through these last 3 slides really quickly, so that there's some time for questions. But I just wanted to mention that, I talked about what we've done in the lab, but there has been some success now with using this approach in clinical trials. So aside from CD5, CD7 is the next common target that's been used in targeting T-cell malignancies with CARs. So on the right, you can see that there's a number of clinical trials being used, mostly with CD7, CD5. Other targets that have been used are CD4, CD1A, TRBC1, CD30, and CD99. CD30 is really, it's specifically for lymphoma. So TALL does not express CD30, but some of those ALCL, that's more seen on the adult side. So there's a group of trials that are really specific targeting CD30, but there's only it's one of those where it's not expressed on all T cells, so you have minimal fratricide. Unfortunately, that target's just not on some of the leukemias that we see and the T cell spectrum, so we've not gone after it more in the lab. CD5 and CD7 seem to be the most common as far as the TLL space. And so this is some early data that we have now. So CD5 CAR T cell trial, this was a trial done by Baylor, where they looked at using autologous CD5 CAR T cells that were unedited. And they enrolled about 17 patients, out of which they were able to expand T cells in about 13 patients. And then 9 of them received an infusion. They did see responses in about 3 patients, as you can see over here, but one of them was a partial response, and there's only one survivor that had, but that they ended up going a bone marrow transplant, and that's what really led to it. But they did show proof of concept that you can use autologous T cells to create a CD5 CAR and see some responses. Now, most of this, they did have challenges because one of the patients did have malignant T cells that incorporated the CAR, so that's always going to be an issue with some of these. But this was really looking at their lymphoma patients, not leukemia, so there's a less chance of having product contamination with lymphoma patients. The figure on the right does show that this is one of the patients that they had a great response from. So you can see this is a PET scan where it shows that all these lesions were able to go away about 8 weeks, except for this one new spot. So they did have a couple of good responses. So, the next step is just to kind of optimize this approach, but it does kind of point that it might be challenging to use an autologous approach in a leukemia patient. This is really more in the lymphoma space where you don't have circulating blasts. And then there's been a couple of trials that have now been published in terms of CD7 CAR T cell results. Now, again, this one was published back in 2021, where again they used donor-derived CD7 CAR T cells. So again, a way to get over that product contamination. But all of these patients with relapsed TLL had undergone a transplant, so they had an identified HLA matched donor, and so they used those patients, the donor, as the T cell source. And this is really promising results where they were able to see that, post-transplant, a lot of these patients did have a sustained response. And even though most of the CD7 negative cells, CD7 positive cells were, eliminated because of the CAR, they were able to have some T cell immunity because there was a population of CD7 negative cells that grew and were able to kind of persist. But the big thing is that these patients had a previous transplant, so the immune system kind of had already been switched up in these patients, and so they really used a donor source to it. But it's really promising work. I mean, several of these patients underwent a second bone marrow transplant with the same donor and were able to then maintain remission. So again, in this case, to prevent fratricide, they used a technology called intrablock, where they blocked protein expression. And finally, I'm going to just mention this. There's this group paper came out about a few months ago, where they used CRISPR base editing, which is a form of CRISPR base editing, where they eliminated the CD7 expression, and were able to infuse this into a few patients. And so this was the first reported success of using a edited CD7 CAR T cell. That the patient then underwent a second transplant and it stayed at remission at 6 months follow-up. So just to summarize, TLA has extremely poor outcomes. There's 3 major challenges, such as fratricide, T cell aplasia, and product contamination. And we've used several strategies to overcome this, and we're definitely showing some early clinical promise. So I apologize, this kind of went a little bit over, so I'm just going to say thank you. These are all the people who work in my lab and have contributed. I'll take a few questions here.
All right. Well, thank you so much, Sunil, for your informative presentation. I know we are getting close to the top of the hour, but we do have time for a few questions, so we'll continue with a few. To our audience, you can still submit your questions. If you would like to, just submit a question in the Ask a Question box located at the bottom of your screen. All right. So, Sunil, our first question asks, what are the pros and cons of using VLR versus single domain antibody, VHH?
Yes, that's a very good question. I mean, we have not. So yes. So overall, now there's a single domain antibody, the VHH approach, which at the time did not exist. So the VLR, since we did not have a superior effect, we kind of went away from that and kind of went back to the traditional approach. The thought was, and the issue with VLR, that the one challenge is always potential immunogenicity, because it's from a different organism that's evolutionarily very distant from mammals. So the single domain antibody, in terms of recognition, maybe not as good compared to the scFv, but in terms of engineering, there's a very similarity because it's a single chain approach. It's just that I think it's really there's a higher chance of immunogenicity with the VLR based approach, since it's not in terms compared to a single domain antibody.
Great. Thank you, Sunil. Looks like we have time for one more question here, so we'll wrap up after this one. Is downregulation of CD5 a concern for causing CD5 negative relapse when treating with a CD5 CAR?
That's a very good question. No, I think that's definitely a challenge. And we know in the CD19 world, really antigen escape relapse has come up as one of the main mechanisms of relapse. And so, there are groups that are exploring dual-based approaches, so targeting CD5 and CD7 concurrently. I think that's where the field will eventually evolve. But this whole downregulation phenomenon definitely raised concern for potentially having an CD5 negative clone escaping and causing leukemia.
Great. Thank you. All right. Well, that will conclude our session for today. So, Sunil, do you have any final comments for our audience?
No, I just wanted to thank you thanks everyone for letting me be part of this seminar, and I hope everyone enjoyed the talk.
Well, thank you again, Dr. Sunil Raikar, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. Now, this webcast can be viewed on demand for 1 year until April 30th, 2025. LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.